So let's first talk about the cell structure and function. So the three topics, I don't know if it's three, cell structure and function that we didn't, that was not on the exam, includes the cytoskeleton for eukaryotic cells, which we discussed. Then we talked, about the different types of junctions that are going to hold cells together in animal cells, which are obviously multicellular organisms. And then the last thing, which I don't have any questions on, is the different types of tissues, but for that part you simply needed to know what the function or role they play in animals. So we'll start with 29, which has to do with the cytoskeleton. So we said that there are three different protein filaments that make up the cytoskeleton. So does anyone remember the name of the three filaments? Microtubules is one of them. Great. Right. Microfilaments. Great. What's the last one? Yeah. What's that? Yeah. Intermediate filaments. And then, so I don't have this part highlighted, but it's just a continuation of this question. Which filament is made from the protein tubulin? So if you think about the name, it's pretty microtubules. So microtubules are made from the protein tubulin. And which filament is made from the protein actin? What's that? Right, microfilaments. So remember, for microfilaments, we said another name for microfilaments is actin filaments, and that's because they're made out of the protein actin. Okay, let's see. Okay, so another topic, the one topic I forgot to mention to you that we covered was protein sorting. So that was also part of cell structure and function. And if you remember, what we're talking about when we say protein sorting is how does in a eukaryotic cell, how does a cell know where to put particular proteins that it's making? So we said that most proteins begin to be made in the cytosol, and that includes proteins that are supposed to be in the nucleus or the ER or the Golgi apparatus. And we talked about how the cell knows to put proteins in a particular area. And what they're showing you here are the different steps of co-translational sorting to the ER. So we talked about the very specific steps of a protein being sorted to the ER. And we said that that happens co-translationally, which means it happens while the protein is being produced. So for 47, you can use the picture. You won't have a picture on the exam or quiz. But can you put the events in order? So those are all the events that happen in co-translational sorting to the ER. Can you put them in order? So what's the first thing, then second thing, and obviously third and fourth?
So is the last one the first thing?
Yes. So the last thing, SRP binds to the ER signal sequence and pauses translation. So that's this step right here. What's that? Right. Then the first one. Then SRP binds to the receptor in the ER membrane. Specifically, it's called the SRP receptor. Right. Then the second, and then the third. So the third step is that SRP is released and translation resumes. And then the final step is that the polypeptide that you just made is going to be released into the inside of the ER, which is the ER lumen. Do you guys have any questions about those steps? Okay. Let's see here. Okay, so the rest of the questions have to do that we have to get to have to do with cell junctions. So given what I told you about the role of tight junctions, what do you think would happen in terms of the different events that they give you there? Which of those do you think would happen if you disrupted the tight junctions that are binding two cells together? And the example that I gave you of cells that are bound together by tight junctions were intestinal epithelial cells that line your gastrointestinal tract. What's that? Right, the answer is C. So remember, one thing that I told you that tight junctions do is that they prevent things from moving between the cells. And specifically, one thing they would prevent from moving between the cells would be the bacteria that's contained within your gastrointestinal tract. And then that's going to prevent it from actually going into the bloodstream and then causing an infection. Okay. So one of the other things that we talked about were different types of anchoring junctions. And so this is asking for the anchorage junction. These anchorage junctions interact with intermediate filaments, and that's on the inside of the cell, and anchor cells to the extracellular matrix. So which anchoring junction that we talked about connects cells to the extracellular matrix, and then on the inside are anchored to intermediate filaments. So if you don't know the answer off the top of your head, you should be flipping back through your notes. So we defined each of them. No. You're close. In terms of the name. It's not E. Getting closer. Right, A, hemidesmosomes. So hemidesmosomes. And then just to point out, you shouldn't know what fibronectin is. So hemidesmosomes, focal adhesions, adherence junctions, and desmosomes, you should all know what those are. Those are the 4 types of anchoring junctions that we talked about. Fibronectin we never talked about. It's actually a component of the extracellular matrix like collagen. So it's something that's on the outside of the cell that's part of the extracellular matrix, but you don't need to know that for this class. Okay. So 67, for heart muscle cells to beat in a coordinated rhythm, ions must move from one cell to the next through which types of junctions? What's that? B? No, not B. Not hemi. No. Oh, yeah, sorry, A. It is A. Yeah, A. Gap junctions. So so I specifically gave you that as an example of cells that are connected by gap junctions. And then 68, you inject a dye into a cell and observe that it moves into an adjacent cell. This would occur through what? Right, gap junctions again. So remember we said that with gap junctions what happens is that gap junctions form a channel that flows, the channel connects the cytosol of 2 cells, and then small molecules and ions are able to freely pass through that channel. So that's a way that 2 cells can directly signal between each other. And so the dye is small enough to move through those channels, and so it goes from one cell to the next. Okay, do you guys see other questions that you want me to go through? Okay. So then the next set of questions we have to go through are the viral structure and reproduction. There's not there's not a very long list of questions. So the first one, number 11, which of the following characteristics do all viruses have in common? Right, A, they're all non-living. So just to go through the other ones, so remember that some viruses have RNA genomes rather than DNA genomes, so that's B. The C, they have a broad host range. That's not true. Some viruses only infect a single species, and then there's viruses that will only infect a single cell type within that species. They all infect eukaryotes, so that's not true because we talked about bacteriophages are viruses that infect bacterial cells. And then the last one, they all contain a capsid surrounded by a fatty envelope, and you know that that's also not true. We said that many animal viruses have an envelope, but we also gave you an example. The bacteriophage does not have an envelope, along with other viruses lacking an envelope around them. So number 12, how do the reproduction of HIV and bacteriophage lambda differ from each other? Right, so HIV encodes reverse transcriptase, while lambda phage does not. And what's the role of reverse transcriptase for HIV?
Turn the RNA genome into DNA that needs to produce.
Right, stuff, yeah. So it takes the single-stranded RNA molecule and makes a double-stranded DNA molecule out of it. Okay, and then number 13, which of the following is not part of the general viral reproductive cycle? No, that happens, and maybe the wording is kind of confusing. So what we described for both viruses that we talked about, for phage lambda and HIV, we said that what happens is some part of the virus binds to a membrane protein that's embedded in the plasma membrane of the cell it's infecting. And so in that case, the membrane protein that it binds to is the membrane protein that recognizes the part of the virus. So you would say that it recognizes that part of the virus. So that does happen in viral reproductive cycles.
D.
Right, D. So reverse transcriptase converts RNA to DNA. So reverse transcriptase is not going to be involved in any virus that has a DNA genome. Okay, are there any other questions that you guys want to go through from this lecture questions list? Okay. So let's move on to the next topic then. And this was actually the topic that your lab covered last week. So we're on membrane function, structure, and transport. Okay. So first of all, in terms of functions, I'm not going to ask you on an exam or quiz to, oh shit, sorry. I'm not going to ask you to list out functions of a cell membrane, so this is just, it's useful to know what their function is. So those are all, it doesn't include all of them, but it does include some of them. And so one of them is another way of stating the first one, and this is the one we actually will talk about in this topic, is how membranes act as a semi-permeable barrier. And so what we mean when we say semi-permeable is that a membrane is going to allow some ions and molecules to get past it, whereas it prevents other molecules and ions from getting past it. So that's what they mean by selective uptake and export of ions and molecules from the cell. Also, in eukaryotic cells, in addition to the plasma membrane, you have the different membranes that surround the membrane-bound organelles. And so that's involved in compartmentalizing the cell. So that's what compartmentalization refers to, the membrane-bound organelles. You saw how membranes are involved in protein sorting. So you saw how the ER membrane was involved in protein sorting. And then in terms of proteins that's secreted to the outside of the cell, you saw how the plasma membrane is involved in that. Also, anchoring of the cytoskeleton is another function. Later on in the course, we actually will get to how membranes are involved in the production of things like ATP and NADPH. Specifically, we'll see how the inner membrane of the mitochondria produces ATP, and then we'll see that the thylakoid membrane of chloroplast produces both ATP and NADPH. And then when we get to cell signaling, we'll see that membranes are also involved with that. Cell signaling refers to how a cell communicates either between cells or how internal signaling happens within the cell. And then it's also involved in both cell and nuclear division. And then also we've seen how the adhesion of cells to each other involves the cellular membrane. And all of these functions, or most of these functions, are performed by the proteins that are either embedded in that membrane or associated with that membrane. And that's a fact that I'll state again when we get to talking about the components of a cellular membrane, one of which is proteins. What gives each membrane a unique function, or most of its unique functions, are the proteins that are embedded or associated with that membrane. So when we talk about the inner membrane of the mitochondria, I told you that it has embedded in it ATP synthase, and that's the enzyme that's a part of that membrane that actually synthesizes ATP. So that would be a protein that's involved in doing that. Okay, so those are just the various functions of cellular membranes. And when we talk about the structure of cellular membranes, we are talking about plasma membranes, which we said in cells separates the outside of the cell from the inside of the cell. But we're also describing the structure. They'll have different compositions, but the same structure. The composition of the membranes that are associated with any membrane-bound organelles inside the cell. Okay. One of the things that you should realize about cell membranes is that they are fluid structures, and we're going to see that most of the molecules that make up the membrane are actually moving about within the membrane. So we talked about phospholipid molecules forming a phospholipid bilayer. The individual phospholipids that make up that lipid bilayer are not held together or held in place. They're actually moving around within the plane of the membrane. And then we'll see that the proteins that are sitting in the membrane are also capable of moving, we'll say, laterally within the membrane. So they can move about within the plane of the membrane. So the membrane has some components of being a fluid because the molecules within a membrane are able to freely diffuse in two directions. So not in all directions like a normal fluid, but the molecules that make up a membrane are able to diffuse or move in two dimensions within the membrane. So the model that describes membranes, the fluid mosaic model, and the fluid part refers to what we just talked about, the fact that the molecules are able to move about within the plane of the membrane. And the mosaic part refers to the fact that so the mosaic part refers to the fact that you have both phospholipid molecules that make up the lipid bilayer with proteins that are embedded in it. So it's a mosaic of lipids and proteins that are embedded within the lipid. So that's just the model that describes the structure of cellular membranes. Okay, so one of the major components of a cell membrane is the phospholipid bilayer. And I've previously drawn this on the board for you. So within a cell membrane, you're going to have two layers of phospholipids. And remember that when I draw, so each of these circles, so this symbol here represents one phospholipid molecule. And we said that this is, I'm not going to write out the full word, but this is the hydrophilic part. And this end is the hydrophobic part of the phospholipid molecule. So that when phospholipid molecules form a bilayer, when they're making a membrane. One layer of phospholipids have their hydrophilic heads pointing towards the water that would be on this side of the membrane. So the hydrophilic heads want to be associated with that, and the hydrophobic tails point away. And then the other phospholipid layer is going to be a mirror image of that. And its hydrophilic heads of the phospholipid molecules that make up this layer are pointing towards the fluid or the water that would be on the other side of the membrane, with its hydrophobic tails pointing towards the tails of the other layer, the tails of the other layer. So now you have a structure where the hydrophilic parts get to be in contact with water and the hydrophobic parts are buried in the center. And if you remember, we talked about the fact that for phospholipid molecules, the hydrophilic head contains a phosphate group along with the glycerol backbone and anything that's attached to the phosphate group. And then the hydrophobic part are the two fatty acid tails that are attached to the phospholipid molecule. So again, the two squiggly lines represent fatty acids. And then the head, the circle part, represents the glycerol backbone with the phosphate group and anything that's attached to the phosphate group. So that's what they're showing you here in this picture. Here's glycerol, phosphate group attached to the glycerol, and then the nitrogen-containing group that's attached to this particular phospholipid molecule is choline. All of that makes up the hydrophilic head, and then the two fatty acids, which are also attached to the glycerol, these two down here, are the hydrophobic part of that molecule. Okay, and then in terms of the parts of the phospholipid bilayer, we'll refer to this inside part of the phospholipid bilayer as the hydrophobic core or interior. So hydrophobic interior or hydrophobic core. When I use that term, I'm referring to the portion of the membrane where all the fatty acid tails are at. So the hydrophobic core is where all the hydrophobic parts of the phospholipid molecules are located. Okay. There's only two new things that I'm going to add from this slide. So the first bullet point is what we just discussed. It's just describing the structure of a phospholipid bilayer. And then we're going to go on to talk about the other components of a cellular membrane in a second. I'm going to add more than two things, sorry. One thing that we're going to talk about, or that I already mentioned, is that the phospholipid molecules that make up this layer are not connected to the other phospholipid molecules in that same layer. They're also not connected to the phospholipid molecules in this layer, but they're not connected to each other in any way. So there are certain movements that are very easy for phospholipid molecules to make, which when I say very easy, I mean that these movements happen all the time, so they occur frequently. So one type of movement that's easy for phospholipid molecules to make is they're able to diffuse laterally. So if you're just thinking in this dimension, this phospholipid molecule can diffuse to the left or to the right very easily. So it can switch places with other phospholipid molecules of the same layer. So lateral diffusion happens frequently. Another movement that happens very frequently is, lateral diffusion is the first one we talked about. The second one that happens frequently is rotation. So what happens in rotation is a phospholipid molecule simply spins in place. So if I was a phospholipid molecule and my head was the hydrophilic part and my 2 feet were the hydrophobic part, then if I just spin in place like this, that's what the phospholipid molecules can do within the plane of the membrane. They can spin on their axis. So that's another one that frequently happens. Another movement that phospholipids can make, but this rarely occurs, is called a flip-flop. So this rarely occurs. There's actually special enzymes that will specifically perform this type of movement for the cell. And what happens in a flip-flop is that the phospholipid molecule in this layer flips and becomes a part of the other layer. And the reason that this happens rarely is because in order for that movement to take place, you're going to have to bring this hydrophilic head into contact with the hydrophobic core, and those two things don't want to be in contact with each other. So that type of movement rarely occurs because in order to make that movement, you would have to bring those two parts into contact with each other. Whereas the other two movements that we talked about do not involve any contact being made between the hydrophilic part and the hydrophobic part. And those types of movements are going to happen very easily and happen often. Okay. Any questions about the types of movements that they can make within the membrane?
So, like, with the fatty acids, is one always going to be saturated with the other unsaturated?
No.
No.
It varies depending on the cellular membrane that you're talking about. And one of the things that some cells, I don't know if I can say all cells, but there are certainly cells that are capable of actually changing the phospholipid composition of their membrane, that's where we're going to talk about next. And one of the things that they can change about the phospholipid molecules that make up the membrane is the number of double bonds that the phospholipid molecules have. So it can put phospholipid molecules in the membrane that have a lot of saturated fatty acids, or put phospholipid molecules in the membrane that contain less double bonds or are unsaturated. And a cell is going to want to make that adjustment if the fluidity of its membrane changes. Okay, so that's where we're actually going now. So we said that membranes are fluid. So membranes have a particular fluidity to them. And cells want to keep their fluidity of their membranes relatively constant. And the reason that they want to do that is if the membrane becomes too fluid, it becomes leaky and lets molecules come in that shouldn't come into the cell. So it becomes leaky if it becomes too fluid. And then if it's too rigid, which would happen if the fluidity decreased, so if the fluidity goes down, then the cell membrane is becoming more rigid. What that can cause, or what that interferes with, is the function of proteins that are actually embedded in that phospholipid bilayer. So you want to keep a certain fluidity of the membrane. You don't want it too high, and you don't want it too low. One environmental factor that will affect fluidity is temperature. And it has the effect that you think it would have. So, well, let's see if you think. So what do you think happens to the fluidity if the temperature goes up? Yeah, it becomes more fluid. So fluidity goes up. And then the opposite happens if temperature goes down. Fluidity decreases. So the organism, one of the things that would cause an organism that's able to change the composition of its cellular membranes, one thing that would cause that to happen would be changes in temperature. So things, so one example is bacterial cells that are not, that are unicellular, that are going to change, their temperature is going to change with environmental changes, and then they can adjust the composition of their lipid bilayers to adjust fluidity of the membrane. And then another thing that affects fluidity of the membrane is the fatty acids that are attached to the phospholipid molecules. Okay, so the fatty acids that are attached to the phospholipid molecules will affect the fluidity of the membrane. And there's two things about fatty acids that affect the fluidity of the membrane. One is length. Okay, and what we're going to see is that shorter tails, so shorter phospholipids with short fatty acid chains make the membrane more fluid? The other thing that affects fluidity is the presence of double bonds. And this you should be able to predict the effect because one thing that we discussed in terms of saturated fatty acids versus unsaturated fatty acids is that at room temperature, which one is usually liquid? Unsaturated. So that would be more fluid than something that's obviously solid at room temperature, wouldn't be fluid at all. So what happens in terms of the presence of double bonds is that more double bonds make the membrane more fluid. Okay, and then the opposite of these two things cause the opposite thing to happen. So whereas shorter tails make the membrane more fluid, longer tails make the membrane less fluid. Okay, so longer tails are going to make the membrane less fluid. And then in terms of the presence of double bonds, if you have fewer double bonds in the fatty acids that are attached to the phospholipid molecules, that's going to make the membrane less fluid. Okay, so just in terms of, I told you that some organisms are capable of changing the phospholipid composition of their membrane. So let's say that you're an organism and the temperature surrounding your cell goes up. What are two changes that you would want to make in terms of the phospholipid molecules in your membrane to keep fluidity relatively constant? So temperature goes up, and so what are two changes that you would want to make about the phospholipid molecule composition in your membrane that's going to keep fluidity relatively constant?
Longer tails or more saturated.
Yes. Sorry, I had to think about it for a second. So when I'm asking that question, you want to think, oh, the increase in temp is going to increase the fluidity. So the organism is going to want to do things that decrease fluidity. And one thing that's going to decrease fluidity is longer tails. So you're going to want to insert phospholipid molecules that have longer tails in the membrane, and that's going to help make the membrane less fluid. So remember, you're trying to make it less fluid. And then in terms of the presence of double bonds, you would want to have fewer double bonds in the fatty acids of the phospholipid molecules that make up the membrane, and that will also decrease fluidity. Everyone feel comfortable with that? Okay. What time it is? Okay. 10 more minutes and then we'll take another 10-minute break. Okay, so if everyone's okay with that, then a third thing that can affect fluidity of the membrane, but not all membranes have this. It's abundant in mammalian cell membranes, is cholesterol. So cholesterol is abundant in animal cell membranes. Bacterial cells lack cholesterol in their membranes, so bacterial cells don't have cholesterol in their membrane. And you can think of cholesterol as a fluidity buffer. So put buffer in parentheses. It's a fluidity buffer. Okay, and the reason that you can think of it as a fluidity buffer is because cholesterol's effect on membrane fluidity is going to depend on the temperature. So cholesterol's effect on the membrane fluid, it has the opposite effect that temperature has on the fluidity of the membrane. So what happens in terms of cholesterol is that at high temps, which would have increased fluidity, cholesterol's effect is that it decreases fluidity. So that's how it would act as a buffer. If there's high temperature, it decreases fluidity, but at low temperatures, it increases fluidity. So again, the effect that cholesterol is going to have on the membrane fluidity is going to counteract the effect that temperature has on the fluidity of the membrane. So again, at high temperatures, cholesterol decreases fluidity, and at low temperatures, cholesterol increases fluidity. Okay. Any questions about that?
Are there any like well-known diseases that affect fluidity of membranes?
Not that I know of. Other questions? Yes.
So on like the layer up there, you said they can basically change position,
Mhm.
go left to right.
Yeah.
Is, like how thick are they? I guess that's my question. Is that how they can get away with it?
What do you mean, how thick are they?
Like we're looking at it, I guess, like two-dimensional. But how many layers typically are there of the?
Are you talking, when you say thickness, are you talking about the distance between this and this?
Like,
Or this way?
I guess behind that way, like how many?
Oh, I see what you're saying.
Yeah.
So how many, well, enough to, I don't know if anyone's ever counted that, but you're talking about the plasma membrane surrounds the whole cell. So depending on the bacterial cell and eukaryotic cells, if we go, just to maybe give you a specific number,
Because I guess if it has to like get out of line to move somewhere else, I guess it could be one minute.
yeah, because so this is showing you how long. So a eukaryotic cell is 7 micrometers long, and a bacterial cell, that bacterial cell is 2 micrometers long. So a micrometer is 1/1,000,000th of a meter. So it's 10 to the negative 6 meters. So it would have to be that long because the plasma membrane surrounds the whole cell. So for the plasma membrane, that's how long and wide it's going to be, enough to cover the cell. And then it's going to differ, and then for membrane-bound organelles, it's going to be smaller because they're on the inside of the cell and they're not as large.
Okay. So like when you say like they can move, like they rotate.
They can, so
Constant then?
it is constant, yeah. And when I described this, I was saying think 2 dimensions because as you're saying, basically in a cell, the layer is not just going from left to right, the layer is coming out of the board and then moving into the board. So this phospholipid molecule can diffuse left and right, but also diffuse out to or into the layers that are behind it. It's just easier to visualize left and right on the board. You just have other questions? Okay. So the next thing that we're going to talk about is, so we're still kind of talking about components and structure of a cellular membrane. And I already mentioned that the fact that proteins are another major component of cell membranes. And the second bullet point is something that I mentioned to you, is that membrane proteins perform most of the specific functions of a membrane. So if a membrane makes energy intermediates, the parts of the protein, the parts of the membrane that actually make the energy intermediates, like ATP and NADPH, are the proteins that are embedded and associated with that membrane. And then also what we'll see is that a membrane's ability to take up certain molecules or to export certain molecules is going to be due to the membrane proteins that are in it. So as one example, there is a specific membrane protein, it's called a transporter, that moves glucose from one side of the membrane to the other. So when a cell wants to take up glucose, it's going to put more of these carriers in its membrane, and then the carriers transport glucose and allow a cell to take up glucose from the environment. In terms of the types of membrane proteins, there's two main types. They're both on the board there. So one type is called an integral membrane protein, and then a second type is called peripheral membrane protein. And the difference between the two is whether the protein is actually embedded in the membrane. And when I say embedded, what I mean is that some part of the protein is actually in contact with the hydrophobic core. Okay, so if the protein has part of it in contact with the hydrophobic core, and we would say it's embedded, then it's an integral membrane protein. If a protein is simply associated with the membrane in some fashion, but it doesn't have any part of it in contact with the hydrophobic core, then it's a peripheral membrane protein. So when you're looking at this picture here, you can see that this protein here has this portion of it that's in contact with the phospholipid bilayer, with the hydrophobic core. So this is an integral membrane protein. And then this protein here, which is associated with this membrane by binding to this protein, is a peripheral membrane protein. So there's no part of this protein that's in contact with this hydrophobic core. So that would be peripheral membrane protein. Peripheral membrane proteins can also be associated with the hydrophilic heads of the phospholipid molecule. So it can also be associated with a membrane in that way. And then so there's not really much you need to know about peripheral membrane proteins. We're not going to talk about any specific ones or specific functions of them. We will talk about specific functions of a certain type of integral membrane protein called a transmembrane protein. So a lot of integral membrane proteins are also transmembrane proteins. And a transmembrane protein, I've defined for you earlier, but a transmembrane protein is a protein that crosses the entire thickness of the phospholipid bilayer. Okay, so what happens with a transmembrane protein, because it crosses the entire thickness of the phospholipid bilayer, you have a piece of the membrane protein that's in contact with the fluid on one side of the membrane, a piece of the protein that's embedded in the hydrophobic core, and then a piece of the protein that's in contact with the fluid that's on the other side of the membrane. Okay, so you would have those three parts to a transmembrane protein. There are integral membrane proteins that are not transmembrane proteins, and as an example, that would be number 4 here. So this part of the protein is embedded in the hydrophobic core, but it doesn't cross over to the other side of the membrane. So this would be an integral membrane protein that is not a transmembrane protein. And then these three right here would all be integral and transmembrane proteins. So another way to think about it, might be confusing for you, so if it is, just forget about it. But all transmembrane proteins are integral membrane proteins. So all transmembrane are integral, but not all integral are transmembrane. Okay. So the definition is up there. They cross the entire thickness of the membrane. In terms of their functions, we'll see in cell signaling that there are transmembrane proteins that function as molecules called receptors. And so a transmembrane protein that's a receptor, what it would do is let's say we'll talk about transmembrane proteins that are in the plasma membrane that are called receptors. And what they would do is the outside part of the transmembrane protein that's on the outside of the cell is going to bind to a molecule that's on the outside of the cells or grab onto it. And then what we're going to see is that's going to activate the receptor, and then the activation of the receptor on the inside of the cell is going to cause something on the inside of the cell to happen. And that's going to be one way that cells communicate with each other within a multicellular organism. They can also function, and we'll get to these two in this topic, as channel proteins and transporters. So channel proteins and transporters are transmembrane proteins. And both of those types of transmembrane proteins are involved in moving something across the membrane. So they're both involved in moving something from one side of the membrane to the other. They just do it in different ways. So they function in different ways, but they do the same thing. Sorry, they have the same function, but they do it in different ways. So same function, but do it in different ways. And then there's also transmembrane proteins that are enzymes. So we haven't gotten to enzymes yet, but enzymes are proteins that speed up chemical reactions. And some of the enzymes of the cell are transmembrane proteins. So those are all functions of some of the functions of transmembrane proteins inside the cell. We'll see each of these functions in the class later on. So in terms of being receptors and enzymes, we'll see that in cell signaling. And then within this topic, we'll talk about channel proteins and then also transporters.
Are lipid anchored proteins just a specific type of peripheral membrane protein?
I would classify them as transmembrane. The lipid part, so what a lipid anchored protein would be is that a piece, so as a part of the protein there'd be a lipid attached to it, and then the lipid part of the protein is in contact with the hydrophobic core. So I would classify it as an integral membrane protein.
Okay.
So yeah, so in addition to having proteins that have carbohydrate modifications onto them, some proteins have lipid molecules attached to them, and the lipid that gets attached to a protein can be involved in having that protein anchored to a cell membrane.
Does it ever okay, so the lipid's pretty much the anchor. Does the protein ever go to the other side too?
No, so that's where. So in terms of proteins that are either associated with the membrane. So I want to say one more thing. It's a little bit over when I said 10 minutes, but one more thing and then we'll take a break. Is that membrane proteins can also diffuse laterally within the phospholipid bilayer. So this transmembrane protein and this peripheral membrane protein are capable of diffusing to the left or to the right or into and out of the board. So that movement can happen for transmembrane proteins, for integral membrane proteins and peripheral membrane proteins. One thing they don't do is this is not going to flip from one side to the other. So this transmembrane protein here is not going to flip so that the inside part is on the outside. And then this peripheral membrane protein is not going to be flipped to the outside. So that type of movement doesn't happen for membrane proteins. But you do need to know that for this class about the two. So for membrane proteins, they're able to also laterally diffuse. So their lateral diffusion, they are capable of that. And then they do not flip-flop, or sorry, I wouldn't say call it a flip-flop, but they flip. They're not flipping within the membrane so that they're reversed. There is a way of keeping an integral membrane protein in place so that it doesn't move. And two ways of doing that. One way is to anchor the transmembrane protein to the cytoskeleton. So this is showing you what you would call an adapter protein. And it's anchoring this protein to something that's on the, in this case, I think they're trying to draw actin. And that would prevent the transmembrane protein from moving within the membrane. And then obviously another way of anchoring a protein in place would be to anchor it to something on the outside of the cell. And one example of that would be to, in extracellular organisms, to anchor the protein to the extracellular matrix, which we said is the scaffolding that's on the outside of the cells that the cells grow on in animals. So you can anchor it to that that's on the outside of the cell, and that can also keep the membrane protein from moving within the plasma membrane, or whatever, or yeah, within this case it would be within the plasma membrane. Okay, do you guys have any questions about those two things? Okay, so we'll take the 10-minute break, and then we'll get started up again.